Hi everybody, this is John, and I am going to talk to you very briefly just about competition ecology and the process that we're going to use the next 5 weeks to study that. The TA meeting and the TA notes I know presented you with a lot of information, so I just put this together to hopefully make you feel a little more comfortable with what's going on. So this is really just about the lab week 1, and we'll get you out of here in 5 minutes. So here's the big picture. To help provide context, in other words, the foundation for the entire lab as it goes through the next 5 weeks, the students are going to need to master a few things. They're going to have to understand what a research question is versus a hypothesis, just bare bones. What are they and how are they different? They're going to have to understand what competition ecology is, or ecological competition, which means they're going to have to understand some of those components, the competitors, their environment, and the resources. And understanding those components then to study ecological competition means they're going to have to understand the role of a model organism because they're limited on time. And then they're going to have to be able to identify some of the factors, in other words, some of those independent variables, and the indicators, the dependent variables, in other words, those things they're going to measure like leaf area and that sort of thing. And then they're going to have to put those factors and indicators into a class-wide research question and a hypothesis frame, and they're going to have to design an experiment that encompasses factors and indicators useful in studying competition ecology. So let's kind of put that in action. So what gets us to this lab report that's due for the students on, was it the 12th of April, I believe it is. We've got all those components, and we really just sort of have to put them all in action, don't we? So let's break it down. Here it is. This is what's going to happen just this week. There's really 6 little subsections that have to happen during lab this week. We're going to ask, what is a research question and a hypothesis? Just come up with a consensus definition so we're all on the same page. Then you're going to ask what competition ecology is. Get everybody on the same page. We're then going to ask what's a model organism. Get everybody on the same page. Ask what are some useful factors, those variables, and indicators of those variables. Get everybody on the same page. And then we're going to start plugging that in to a research question and a hypothesis. And again, we're going to get everybody on board in your class. And then finally, we want to be able to operationalize all of this by coming up with some sort of experiment. So what does that mean? Well, let's break that down even further. So right off the bat, the first thing you're going to do in lab this week is ask the students, what is a research question and a hypothesis? What's the difference? They're going to do some small group brainstorming, and then they're going to come together, come up with a large group consensus. Once we've got that down, the next thing you're going to do is ask, well, what is competition ecology? Same format. Right, we did some small group brainstorming, some large group consensus here. Now we can start to put that in use. Well, what is competition ecology? We'll do some small group brainstorming, large group consensus. Again, have them write these answers on their whiteboard, and then maybe you can come together and put them all up front, come up with a large group definition for what is competition ecology. Once we have these two down, then the third thing you're going to do is ask, well, what is a model organism? And it's going to be the same process. Small group brainstorming, large group consensus. We can start to build on this then, and asking how are we going to study that? Again, small group brainstorming, large group consensus, which then allows us to start breaking this down even more. Well, what is a model organism? They'll come up with some small group answers. You come together as a class and come up with one big consensus. Once we do that, then we can start to ask, well, this is about the environment, right? What are some factors or variables that we want to test, and what are some indicators of those variables that we're going to test in our model organism? It's the same basic thing, right? Small group brainstorming, large group consensus, and you'll see, right? These just all fit into one another. So that by the time we get here, we're ready to do, again, some small group brainstorming, large group consensus. And again, the TA notes go into this much more in depth, and that's part of why they seem so heavy duty. Once we've done that, well, now we can bring in another portion for the lab, right? Once we've done these 4 things, we're now set to start asking, what can we do as an overall research question, and what can we have as testable hypotheses for each of the tables? But it's the same basic thing, right? Small group brainstorming, large group consensus that then feeds into the next part. So what we're really doing is building up all these pieces that are going to allow us to do this, so that ultimately we can do some small group brainstorming, large group consensus to come up with a class research question and a hypothesis. Once we've done these 5, then we finally get to how do we operationalize the class research question and the hypothesis. What's going to be the experimental design that we're going to do? Same basic thing. You get the idea, right? To be able to ask what our research design is, same kind of thing. Small group brainstorming, large group consensus. So what gets us to this? Ultimately, if we step out maybe to the 40,000 foot level over the next 5 weeks, what we'll see is this week then, those 6 things we just pointed out are really here. This is ultimately to get us to some sort of experimental design, so that the following week then, when we do the cheatgrass case study, we can refine this and we'll actually be able to plant. 3rd week then, we'll actually do some initial data gathering, and the students then should be able to write their intro and methods at a minimum. The following week is spring break, so Jean and I will just be doing the watering then. Week 5 is really the kind of the big push. They're going to try to gather most of their data, and then they're going to start the results and discussion sections, so that this they can then turn in right around, I think it's the 12th of April. All of that then means they're not turning in that lab report at the end of week 1, right? All they're going to do is turn in the design sheets for their experiment and their planting schematics. Those are both in the lab manual. And then Jean and I need a list of materials so that we can have everything ready for the students to plant when they come back the 2nd week of this lab. So in the end, right, all the steps, there's just 2 deliverables, and we're walking the students through several baby steps to get them there. And in doing that, you're simply helping them develop the components through small group brainstorming, and then they come together and they do some large group consensus building for each of those smaller components. So small group, big group, small group, big group. That's really the brunt of it all. And I would say thank you for listening. If you have any questions at all, please don't hesitate to stop in my office or Jean's office anytime, even if it's during lab. It's fine to step out and just sit down and say what's going on, and we'll see if we can come up with some sort of answer for you. So please don't hesitate to stop in and talk. We'd love to chat with you. Be well. Thanks for listening, and have a great day. Thanks.